Hello there, my name's Dr. Adam Turner. I'm a senior lecturer at Swansea University and I'm also a fully qualified pharmacist. Today I want to talk to you about the way that nature has impacted the way that we discover medicines. I'll give you lots of examples of how different plants and animals have helped us to produce some of the most effective medicines that we have today. We'll think about drugs that you may be familiar with, but also some that you won't be. But all of them have links back to nature. The first example I want to talk to you about is the hungry goats. According to popular legend, maybe 1,000 years ago, an Ethiopian goat herd named Kaldi saw his goats dancing and messing around. Kaldi had previously found the behaviour of his goats to be irreproachable, grumpy even, so he knew that something unusual was going on. When Kaldi investigated, he saw that the goats were merrily eating the red berries and shiny leaves of an unfamiliar tree. Kaldi decided to try some, and when he did, he joined the dancing goats and became the happiest herder in happy Arabia. Some time later, a passing monk observed Kaldi and the goats. When Kaldi told him about the berries, the monk thought they might be the answer to his prayers, literally. It seems that the monk was always falling asleep in the middle of prayers. When he ate the berries, he stayed awake. The unnamed monk came up with the idea of drying and boiling the berries to make a beverage. His fellow monks loved the new drink because it encouraged them to pray, and it tasted good too. The drink they discovered was coffee, and the drug causing the high was caffeine. This is one of the earliest stories where nature has given us a compound able to change the human body. One thing we all probably know from medieval medicine is the use of maggots and leeches when treating patients. Maggot therapy involved the use of maggots of the green bottle fly, which are introduced into a wound to remove dead or infected tissue. In simple terms, the maggots physically feed on dead tissue and release special chemicals into the wound that break down dead tissue into a liquid form that the maggot can easily remove and digest. During this process, they also take up bacteria, which are then destroyed within their gut. This process is so effective that maggots can often clean a wound within a few days. This method is still used in modern medicine by doctors and specialists who have found that maggots are able to cleanse wounds much more rapidly than regular bandages. While maggots should not be regarded as a cure for all types of wounds, by removing dead tissue and any associated bacteria, in most instances, they will improve the condition of a wound and allow the process of healing to begin. The leech's use in medicine was documented almost 3,000 years ago in the tombs of Egyptian pharaohs and is described in texts from India, China, and ancient Persia. In Western medicine, medicinal leech peaked in the early to mid-19th century. About 100 million leeches were used each year in the 1830s, with a typical treatment requiring a dozen or more leeches. Leeches are sometimes used today. An example would be when severed body parts, such as fingers, are reattached. One of the most challenging parts of this surgery is the rejoining of veins. Injured tissue can swell, which can reduce blood flow and prevent healing. Leeches help to drain the excess fluid, allowing the tissues to re-establish their own blood supplies and to start healing. The leech's secretions may also help blood flow to the site of the leech bite and, as a result, also to the surrounding tissues. Leeches have helped save thousands of severed fingers, toes, noses, and ears in recent years, and have also been increasingly used in breast reconstructive therapy. In 1848, gold was discovered in California. The following year, most of the people who were heading west were gold prospectors. Much of the gold found by panning. The gold was washed out of the mountain rocks by the streams. It mixed with the gravel on the riverbed and made a fortune for those looking for it. Bioprospectors are modern-day equivalents and share the same passion for finding something valuable in nature. Bioprospecting is the exploration, extraction and screening of biological diversity and indigenous knowledge for commercially valuable genetic and biochemical resources. Simply put, it is the discovery of drugs in nature. In the early stages, discovery largely focused on the plants from forests. However, in recent times, various other candidates like insects and animals have been explored with considerable success. It's some of these successes we'll be celebrating today. Plants have been used as medicines for centuries, often without any noticeable benefits. Plant materials were processed for medicinal use by chopping or grinding into powders and through mixing to make teas or for smoking or chewing. Often mixtures of plant products were combined to create the most beneficial medicines. While the majority of these products were not effective, it was the best that could be offered at the time. But, despite some preparations lacking noticeable benefits, some have given us useful products when they have been further investigated. Aspirin is a modern miracle medicine and one of the most widely used drugs in the world. It's extracted from the bark of the willow tree, which was recommended for the treatment of aches and pains by the ancient Egyptians in the Ebers Papyrus. People used to chew twigs to alleviate pain, and it's still possible to buy willow bark for this purpose. However, most of us take over-the-counter aspirin tablets, where the active ingredient, salicylic acid, has been replicated in a laboratory. The chemical salicylic acid was extracted in the 1830s, both from the willow and also from a plant called queen of the meadow, or meadowsweet, whose Latin name is Spirea ulmaria. Later, a chemist working in a laboratory in Germany changed the chemical makeup of this to make it less irritating to the stomach, and called his new product acetyl salicylic acid. Its common name became aspirin, where the A referred to the acetyl part and the spirin to the Spirea ulmaria, the source of the salicylic acid at the time. Aspirin was one of the first modern drugs to be synthesized and may be considered the foundation of today's pharmaceutical industry. Morphine was discovered by a chemist from the opium poppy, a plant that has been the source of pain-killing drugs for almost 5,000 years. The Sumerians were thought to have first grown opium poppies, which they called hulgil, or joy plants, in around 3000 BC. It is still the drug of choice in many cases of severe pain. By using the morphine structure as a model, chemists were able to create stronger pain medicines in the laboratory. The Madagascar, or rosy periwinkle, has had a long history. During World War II, it was used to help treat diabetes. After the war, extracts of the plant were investigated by chemists, and it was found that the flower actually caused damage to white blood cells. When this was discovered, chemists considered how the Madagascar periwinkle might be used in white blood cell cancers. 4 compounds were discovered in the 1960s, and 2 of these have become some of the most effective chemotherapy agents available today. One, called vincristine, has revolutionized the treatment of childhood leukemia, turning it, when used together with other cancer medicines, into a disease that may be cured in many patients. However, Production of these chemicals was complex and not fully understood by scientists, so doctors had to rely on crops of the plant itself to treat these deadly diseases. It took about 5,000 kilograms of dried leaves to produce 10 grams of the drug. To feed their cows, farmers in North Dakota were forced to grow sweet clover, which had been introduced to North America centuries earlier from Europe and Asia. The use of clover fed the cows, but came with considerable consequences. Cows began to die, either from minor bumps or cuts that led to unstoppable bleeding, and other situations where the farmer could not stop the bleeding of the cows. Publications about this became known as sweet clover disease. Veterinarians noticed it was only when cows that ate spoiled sweet clover hay that got the disease. Years later, a chemist discovered that sweet clover and the mold growing on old hay was responsible for the disease. Warfarin is a synthetic compound derived from this discovery. It is a drug used for the long-term prevention and treatment of blood clots. But how do we know which plants might have the right chemicals in them to be used as drugs? Well, studies have shown that brightly coloured bugs like to sit on these sorts of plants. Trying to find the right type of plant is like trying to find a needle in a haystack. Plants do not advertise the fact that they contain potential medicines. The insects that feed on them, on the other hand, do. Insects eat these bioactive chemicals from the plants and advertise the fact that this makes them poisonous to predators with their bright colours. Many insects signal that they are poisonous to predators with flashy colours, often bold reds and yellows. Many tropical insects carry toxins to protect themselves from predators. In fact, in the early days of drug discovery, researchers tried collecting insects in order to extract their active compounds. However, the amount of insects needed would be huge. The insects often do not produce the toxins themselves. Instead, many eat toxic plants and then secrete the poisons. As a result, the search for new drugs inspired by natural compounds now focuses on the plants rather than the insects. We've also been able to discover drugs within the animal kingdom. The human body has systems in place to maintain blood pressure. Central to this system is an enzyme called ACE. The action of ACE in the body increases blood pressure. In 1949, a scientist discovered that when the venom of a pit viper got into animals' blood, there was a life-threatening plunge in blood pressure. This helped scientists realizing that inhibiting ACE might be an effective means for treating human high blood pressure. Further investigation led to the synthesis in 1981 of captopril, the first ACE inhibitor that could be taken by mouth. Numerous ACE inhibitors are now available on the market and are among the most effective and best tolerated anti-high blood pressure medicines available. The venom from snakes has inspired other scientists to look at more poisonous creatures. The toxin obtained from the Trinidad chevron tarantula venom is being considered as a new way for treating pain. Extracts from the Central Asian spider venom have also been studied for pain treatment. And toxins isolated from the Australian funnel web could be used to protect the brain from injury caused by low oxygen. Next, we'll look at scorpions. There are some cancer surgeries and some parts of the brain where tumour tissue and healthy tissue look very much alike. In these cases, a doctor can use the view of the brain where the tumour is illuminated in fluorescent green alongside the healthy tissue. This method helps surgeons understand how much tumour they've removed and how much is left behind. The green glow comes from a drug derived from a potent venom of the Israeli deathstalker scorpion. The drug uses the venom and an infrared dye to seek out and illuminate tumours of all kinds, including in breast, colon, and skin. Exnantidine is a drug based on the saliva of the Gila monster lizard and has been used in patients with Parkinson's disease, showing beneficial effects on nerve cells by slowing down or stopping the processes. Cone snails defend themselves and paralyze their prey, worms and fish, by injecting a cocktail of toxins through a hollow, harpoon-like tooth. Each of the estimated 700 species is thought to make as many as 100 to 200 different toxins, so there may be as many as 70,000 to 140,000 toxins in all. But even though less than 1% of the likely total have ever been investigated, there are several potential new medicines that have already been identified. The most important of these are for treating pain. One toxin, which has been used to create ziconotide, blocks a specific type of nerve cell that carries pain impulses to the brain. There is evidence that this drug is as much as 1,000 times more potent than morphine, but does not have the same addiction or tolerance. The use of snail toxins for pain represents a significant change in the history of pain management, which has been centered on opioid-based medicines for centuries. Drugs discovered from the Caribbean sponge act so that cancer cells cannot divide. Also, the Okinawan sponge could serve as a useful chemical for future anti-malarial drugs. Some of the compounds from marine life forms are extremely potent, given that these organisms have to defend themselves from attacks in vast volumes of water that dilute the compounds. A product extracted from a species living off the Mediterranean Caribbean Sea has been considered as an anti-cancer molecule for ovarian cancer. The final story I want to leave you with is about gastric brooding frogs, the only amphibians known to raise their young in their stomachs. They were discovered in the 1980s in undisturbed rainforests in Australia. The female swallows her fertilized eggs, which then hatch in her stomach. When the hatchling becomes fully developed, they are delivered to the outside world, propelled by their mother's vomiting. They then continue to develop into adult frogs. The stomachs of most species, including frogs, contain cells that create acid, and these are what help to digest food. There are also compounds that stimulate the emptying of the stomach so that its contents can be moved along into the rest of the digestive system. When we take in food and other things into our stomachs, we release these compounds. Preliminary studies with gastric brooding tadpoles demonstrated that they secrete a substance, or substances, that stop the production of the acids and then stop the stomach emptying, so that they do not end up being digested by their mother. But these studies, which might have led to important new insights for treating humans for things like excess stomach acid or peptic ulcers, something that can affect millions of people around the world, could not be continued because the species of these frogs became extinct. We need to look to nature for a lot of the drugs that we develop. So we need to look after nature and ensure that we're doing our part to protect the diversity so that we may find the next big breakthrough drug to help us move forwards as the human race. I've been Dr. Adam Turner. I thank you very much. I've put my email address on this slide so that you can get in touch with me if you've got any questions about anything that I've talked about in this presentation. Thank you to Merthyr Science Festival for inviting me to speak. Thank you to you, and goodbye.